Good afternoon or evening, depending on where you are. My name is Matthew Cobham, and I'm responsible for lighting application at Philips Lighting. And on behalf of the Philips Lighting University and Philips, I would like to thank you for joining today's webinar entitled Evolution on a Rotating Planet, the Importance of Biological Clocks. Now, this is the third in the series with the theme Light and Wellbeing in collaboration with Parsons the New School for Design. And I would like to introduce you to our very eminent speaker today, Professor Serge Daan. Before I hand you over to him, I would like to give you a little bit of background. He studied biology at the University of Amsterdam. He was trained as a postdoctorate by the two founders of modern chronobiology, Jürgen Aschkoff at the Max Planck Institute for Behavioral Physiology, and Colin Pittendry at Stanford University in California. His further career was at the University of Groningen in the Netherlands, where he became professor and finally dean of the Faculty of Mathematics and Natural Sciences. His research interest has always been focused on the temporal organization of behavior in animals and humans. Dan is often invited to conferences as a keynote speaker, and that even continues during his retirement. And he's been awarded many distinctions, including the Victor and Erna Hasselblad Award in 1982, the Alexander von Humboldt Research Prize in 1992, the Japanese Eminent Scientist Award in 2008, and most prominently, the International Prize for Biology in 2006. He was elected as a member of the Dutch Society of Sciences in 1999, Fellow of the Royal Society of Canada in 2000, and received the knighthood in the Order of the Dutch Lion in 2004. So, just before I hand over to Serge Daan, I'd like to also mention to you that if you have any questions, please note those down during the webinar, and we will provide you with an email address at the end where you can send those to. Without further ado, I would now like to give a very warm welcome to Professor Serge Daan.
Thank you for that very kind introduction, Matthew. Good morning or good evening, everybody, wherever you are on this planet and whatever time it is for you. Each one of you is deeply aware of the alternation of night and day. Night and day are the result of the rotation of the Earth around its axis. Night and day profoundly affect each one of us. Once per day, human beings change from the vertical position into the horizontal. If you would be able to observe this from outer space, you would see a wave going over the globe, a wave like in a soccer stadium, a wave that closely follows the transitions of dawn and dusk as these move over the planet, a wave that continues year after year, century after century, with amazing precision. A wave in which we are joined by thousands of other species that change from activity to rest and vice versa once every 24 hours. Night and day have been present ever since the beginning of life on Earth. The rotation of the Earth very gradually slows down, only with half a millisecond each century, but it has always been there. Night and day have a profound impact on the evolution of life. In my talk, I will discuss with you some of the basic phenomena in the living world to which this rotation has led, and some of the key elements in the adaptation to night and day. In doing so, in discussing these issues, I have decided to divide up my talk into three different parts. I will first talk about common principles in biological clocks, which have evolved as an adaptation, and then talk about the diversity of functions observed by these clocks. And finally, I will discuss the role of so-called circadian pacemakers in the central nervous system. The story of the study of rhythm starts actually with a particular plant, the touch-me-not, or the sensitive plant, in which already in the 18th century people have been studying the movement of the leaflets. If you look carefully, you can see that the leaflets are connected to the main stem of the leaf by little joints. They are seen here as white or yellowish dots, and these joints make the leaflets move once per day from this open position in daytime to a folded position at night. And it was this already attracted the attention of many scientists, but also of Charles Darwin in the late 19th century. And Darwin was the person who advanced the hypothesis on why these plants do that. He suggested that they reduce radiation and heat loss at night when it's cold, and by folding up the leaves and opening them again for photosynthesis in daytime. Now, the person who made the major breakthrough in the study of these leaf movements was Augustin de Candolle, a Swiss botanist, and he studied these movements in continuous light, where he exposed the plants to continuous light. And he writes about when I exposed sensitive plants to continuous light, he showed the normal alternation of sleep and wakefulness. But each cycle was a little shorter than usual. The acceleration was often 1.5 or 2 hours per day. And then he concludes, on the basis of these observations, I think one can conclude from these facts that the movements of sleep and wake are caused by a disposition for periodic movements that is inherent in the plant. Now, that was a very profound insight. De Candolle realized if the period becomes, if the rhythm is visible and the period is not 24 hours, it cannot be generated by the rotation of the Earth. So the only solution is that it comes from within the organism itself. Today, these facts are known for many different systems, for hundreds of different species, both plants and animals. Here, for instance, you see results from a Syrian hamster, and you see exactly the same as what de Candolle observed. I have to explain the so-called actogram here on the right. This is a recording of the wheel-running movements of a hamster in a cage. You see here at the end of the light period, every day that the hamster starts running in the wheel, that is being recorded. But when the light is completely turned off on the 7th day and remains turned off, the rhythm goes on with remarkable precision from day to day, but the period is no longer 24 hours. It shifts forward by about 1 hour per day. So it is a period of 23 hours, and that cannot be generated by the rotation of the Earth. It has to come from within the hamster. Now, researchers have found it very hard to believe that organisms would be able to generate a rhythm from within, and that they would be so sophisticated so as to know what time of day it is. So people suppose there would be some unknown environmental factor, a factor X, that was responsible for this. And the studies in the early 20th century were many of them were dedicated to the search for this factor X. And people put organisms, plants and animals alike deep into salt mines to the high Arctic, where it's continuous daylight. There was even an experiment with table mounted on the South Pole, which rotated in the opposite direction from the Earth, so that the table would be stable in space. And then experimental setup was made on top of that table. And finally, eventually in the 1980s, there was an experiment in outer space to bring fungi out of the realm of the Earth's rotation at all. And in all these cases, the rhythm continued. And people then concluded that it could not be that it came from the rotation of the Earth, quite correctly so. But they could have spared themselves all the trouble, because the simple fact that the period is different from 24 hours was sufficient to come to that conclusion. Now, the field was very diffuse in the early 20th century, and people working on different organisms, on fruit flies and on plants, and they had rather little contact with each other, and they were not considering this as the same, essentially the same system and the same problem. And that changed when in 1960 there was a major conference organized, the Cold Spring Harbor Symposium of Biological Clocks, and that was organized by two people, Colin Pittenrick in the United States and Jürgen Aschoff in Germany. And these were also the two leading scientists who unified the fields and the studies on biological rhythms, and who pointed out that there were very general principles followed by organisms. They proposed that essentially there is an endogenous self-sustained oscillator with a period of about 24 hours, but not precisely, and that generates a so-called free-running rhythm in constant conditions. But when in the normal situation that oscillator is connected via a light sensor, via the eyes, to the light-dark cycle, the light-dark cycle will transfer that information and generate what is called entrainment, synchronization of the oscillator by the light-dark cycle. So, and that is how animals and plants are organized, was the proposition. And if you remove the eyes, or you put animals in constant darkness so that they do not see the light-dark cycle, they will show the free-running rhythm. That has been demonstrated in, as I said, in many, many species. I'm showing you two particular examples from Aschoff's work in Germany. This is on chaffinches, and they also can be easily recorded, the activity and rest. You see again the spontaneously recorded movements by a switch underneath one of the perches in the cage. And you see that this chaffinch shows in constant darkness the so-called free-running rhythm, but as soon as a light-dark alternation is installed in the cage, then it gradually moves to complete entrainment and synchronization by the light-dark cycle. Under such conditions, the activity onset is here preceding lights on by 1 or 2 hours. So it cannot be a response to the light, because the light is only later than the activity onset. That beginning of the activity is coming from within the organism itself. And there's a gradual change, a gradual movement towards that stable phase relationship, which is very much like what we experience when we fly over the globe, and when we experience in the week afterwards some kind of jet lag. You see that also in the right-hand side in another chaffinch, which is now, it's not exposed to continuous darkness or continuous light, but it's exposed to a light-dark cycle with 12 hours of light and 12 hours of darkness. And that light-dark cycle is shifted over 12 hours as though the animal is flying from Amsterdam to Wellington in New Zealand, 12 hours away, and then gradually the rhythm shifts to the new phase position. So that is what entrainment really is about. But how this works, how the light does this, is not solved by that simple observation. So what Pittenrick in particular contributed to the study of entrainment is that he used brief light pulses given at different phases of the daily cycle in constant conditions. This is done here in a hamster, but they are measured. Such measurements have been made in many different systems. The hamster has its very precise endogenous daily rhythm. It's called a circadian rhythm because the rhythm has a period of about a day, circa ds. Once every 14 days or so, the hamster is exposed to a very brief light pulse of 15 minutes. And such a brief exposure to light can shift the phase backward or can shift it forward with phase shifts up to 3 hours by that brief light pulse. And whether it is shifting backward or forward is dependent on the phase in the rhythm where the light is being applied. And if you plot these shifts as a function of the phase, you get a so-called phase response curve. Phase response curves have, as I said, been measured for many plants and many animals, and you would think that maybe this has to do something with whether the animal is nocturnal, night active, or day active animal, but it makes no difference. You get the same general shape in regardless what type of animal it is. And that it has this particular shape has great importance because this is the way in which the system can self-correct for its aberration from the rotation of the Earth. So if you see here the phase response curve with delays followed by advances and then followed by a part where there's rather little response to light, you can understand that when the system, when you're now looking at a slow rhythm or a slow pacemaker or a slow clock, it will tend to drift into the day with this sensitivity, and it will thereby generate a correction, a phase shift forward. And a very fast clock, which is faster than the rotation of the Earth, has an inbuilt correction mechanism to shift backwards and to synchronize in that way. And that is the way in which the whole living world keeps in sync with the rotation of the Earth. How about humans? Jürgen Aschoff, a medical doctor by training, but a medical and a human physiologist. He was the director of the Max Planck Institute of Behavioral Physiology south of Munich in the Bavarian foothills. And he made in that institute of his an underground bunker. You see here the entrance to the bunker, and you see here a plan where there were two separate units where people could live in isolation. Volunteers were spending there in most experiments 4 weeks in a row without any contact with the outside world, without any access to clocks or so. And they were being studied for their spontaneous rhythmicity. And those experiments have been going on for 25 years. Altogether, more than 400 experiments have been done there. And the general pattern observed is just like in animals, but most humans have the tendency to have a slightly longer period than 24 hours. It's more closer to 25 hours, and you see that the onset of wakefulness day after day is drifting backwards, and the onset of sleep likewise. About in the same ratio is being maintained, and typical over the course of 4 weeks, there's one cycle less than 28 days is being made, because each cycle is a little bit longer than 24 hours. And then when the experimenter comes in, then the subject usually says, but I thought we had agreed on 28 days, and I have made only 27 so far. Because the subjects are not aware that their clock is running a little bit slower than the rotation of the Earth. But that is the case in most cases, most individuals. They also adhere typically to 3 meals a day, and that is of interest in another case, which I'll show a little bit later in the talk. So humans have the same tendency to do this. Do they also respond in the same way to light? That is a question which was already basically addressed by Aschoff in his studies, but we have now recently much more detailed information on that due to work by Till Renneberg, especially a professor at the University of Munich in the same area, and also a former collaborator of Aschoff. And he designed a questionnaire which can be filled in over the internet. That is the so-called Munich chronotype questionnaire, where people are being asked a simple question: when do you go to bed? And when do you turn off the light and fall asleep? And same thing in the morning. But Till was, and then he can calculate from that the midpoint of sleep. But he was clever enough to realize that the daily work schedule has an influence on this, of course, so he asked the question for work days and free days separately. And what comes out of there is that you have a pretty broad distribution, but with a clear average time, and the mid-sleep on free days is somewhat later than that on work days. No wonder. This is from some time ago. At that point, the number of entries to this very popular website was 60,000. It's now more approaching, I think, more like 200,000, but the basic phenomenon does not change. But it is so popular that Till could calculate the midpoint of sleep on free days for different longitudes all across Germany, and eventually also we have data from the Netherlands with the same system. And you see that people in the east of Germany and towards the west of Germany sleep at different times, and their mid-sleep time moves from going from east to west with about 4 minutes every degree of longitude, and that is as much as the sun moves from going from east to west, because 4 minutes per degrees longitude gives you 24 hours for 360 degrees. So humans, with their sleep timing, with their spontaneous sleep timing on free days, follow the sun. And that's the upshot of this, and the most recent data expand this. This is essentially the wave I was talking about in the beginning of my my presentation, the wave is being made visible, and today these data are expanded into the rest of Europe and even to many other places in the world. So we follow the sun, so we are just as sensitive to light as animals. And we now know also from various studies that humans have a phase response curve for light, which is not different, not basically different from that which I have shown for small mammals. And not only humans have this phase response curve, also fruit flies have it, and also paramecium, a single unicellular alga which lives in the water. And just any organism living on the earth and evolved on the earth has this same basic mechanism to keep in sync with the rotation of that earth. There's unity also in the organization. And now I'm talking not about plants, but only on animals. Animals have pacemakers, central circadian pacemakers in their central nervous system. And these, as predicted by Colin Pittenrick in 1960, these pacemakers are connected to eyes and to photoreceptors. In birds, for instance, it is the pineal on top of the brain, which in sparrows perceives the light through this very thin skull. And that is sufficient to reset the pineal, and the pineal controls the physiology of the body. In snails, it is a group of neurons at the bottom of the stalked eye. In cockroaches, it is a structure just behind the compound eyes. In mammals, including humans, it is the suprachiasmatic nucleus, a small, two small nuclei at the bottom of the brain which receive immediate input from the retina. And that is the area where Russ Foster in his last webinar has been talking about how that light is perceived by the eye there. So in all cases, we know there is a central nervous pacemaker, a circadian pacemaker, which is involved in the generation of rhythmicity, and which is connected immediately to photoreceptors. There's unity also in the mechanism. I'm not going deep into the molecular biology of the system, but just briefly tell you that much progress has been made in the molecular mechanism of circadian rhythms. It's generally within the cells. It's based on two types of proteins. There is a Pas protein complex, which is involved in promoting the transcription of DNA, so that messenger RNA is being formed that is released from the nucleus into the cell, and that is then translated into a protein. And these clock proteins, they return to the nucleus and they inhibit their own transcription. And those two movements generate the pattern of rhythmicity of the cell. That is where the circadian rhythm is being produced. And that same system we find in fungi, in molds. I'm not going to discuss the details of it, but it's in rhythms in fungi follow that principle. In fruit flies follow the same principle. The clock proteins and the Pas proteins are different between these all these groups and species, but they have all the same, essentially the same setup of this mechanism. And the last example is from mice. So there is unity in the response, in the behavior, the response to light. There is unity in the physiological organization with pacemakers, and there's unity in the molecular mechanism. So they have biological clocks do follow common principles to keep in sync with the rotation of the Earth. But the functions of these pacemakers are very diverse. And the diversity is so large that, of course, that is not coverable, but I would like to highlight a few things about this. I will first show you what is probably of very general importance, that is the matching of the internal cycle with the external cycle. That is one basic function. There is orientation in time, how animals measure their day lengths in the course of the season to adjust their reproduction to time of year. There are daily routines for individuals. There's the internal synchrony of physiology. And these are all too complex now to discuss here in detail. But I would love to show you a little bit on the timing of critical phases in life, on sun compass orientation, and then the rest of the talk will be dedicated to activity arrest. So the matching of cycles there's a very interesting study on a very primitive form of life. This is a prokaryote, which doesn't even have a cell yet. It's a blue alga, cyanobacterium. And one can, what a group of researchers, particularly Takao Kondo in Nagoya in Japan, have been doing is building in reporter genes which make these organisms glow when in a certain phase of the circadian rhythm, so that it can be made visible. And also what these researchers have done is generate mutants where the period is different from 24 hours. It is in the upper one here, it is, for instance, a period of 22 hours, so it's shorter than 24, and the lower ones have periods of 28 hours. They have very slow rhythms. And what Carl Johnson and his colleagues have been doing in the United States is let these mutations compete with each other under different circumstances. And here is the bottom line figure of what happens if you mix the 24-hour mutant strain with wild type. The wild type has about 24 hours and the mutant strain 28 hours, and you can expose them to either constant light, and then they do not change over the generations. They maintain a 50-50 contribution. Both of them are equally strong in competition under constant light. But if you put them in constant, in a light dark cycle of 24 hours, 12 hours light, 12 hours darkness, then the P28 mutant disappears within 40 generations. It cannot compete with the other one. If you put them in 30 hours, 15 hours of light followed by 15 hours of darkness, then the wild type cannot compete, and it is the mutant which remains. So what the system does is make the organism match the cycle in the environment, because that gives the best chances of survival. Critical life events is fun. There are lots of examples of it. The most famous example is of Drosophila, which emerges from its pupae. The adult fly emerges from a pupa And Pittendrick has used that system extensively, and he has recorded the time at which this happens in populations of pupae. And you see a very, very precise rhythm which continues in constant darkness. And here are, in the lower part, are the average points. You see how precise this rhythm is. This is only once per lifetime that this happens, but in the whole population, it is a rhythm. And it occurs at a specific time, especially early in the morning. And what makes this such a critical time, the best time to do this, is not quite clear. The suggestion of Pittendrick has been that it's the air is most humid early in the morning and gives the best chances for eclosion of the adult. That has not been completely resolved, so I now go to tell you a little bit about another system where we know at least what happens, although the data are not very extensive. I like to talk about this because I've done this myself a long time ago on the island of Spitsbergen, where I did some field work on the guillemot, Brunig's guillemot, which breeds in large numbers on the rocky slopes of on the coast of Spitsbergen. And we, together with my friend and colleague Joost Tinbergen, we made a study on the jumping of the young guillemots off the cliff. This is once in a lifetime that this happens. The young has the adults, they lay only a single egg, and that egg hatches and the young grows up. And then after 3 weeks, it has grown sufficiently to jump into the water from the cliff, but it cannot fly yet. And the cliff is 450 meters high, so it's quite a jump. And let me see here. Oh, yeah, let's go back here. You see the rocky slopes. It's about 450 meters from the bird cliffs to the sea, and it is about 450 meters high. So it's a 45 degrees slope which the young have to make in order to reach the sea. And it's pretty dangerous because if they fall on the ground, they will are most likely to be eaten by predators. They do not die from the fall because there's vegetation down there. And the interesting thing is that they do this in the middle of the night, or just before midnight especially, and nothing in the middle of the day. And the even more interesting this is if you realize that this is at Spitsbergen 77 degrees north, and it's continuous daylight. So it is not so that the night is dark and thereby safe. No, the night is safe because there are large numbers, and they are doing this together, and they generate safety in numbers. And we quantified that, and it's quite clear that the ones which of these young ones which drop on the ground have a great chance of being killed. Certainly in the beginning of each peak and towards the end of each peak, but in the middle of the night they have a chance to escape. So that makes it critical, and that makes timing of great importance for this particular event. So that's just for the fun of it. Also fun is the role of the biological clock in orientation towards the sun. Many animals use the sun as a beacon for their orientation in space. And one particular example, and an impressive example, is that of the monarch butterfly, which breeds in all through the United States, that in the eastern populations, in the winter, they migrate to a single piece of woodland in Mexico where they all collect all together. And in order to get there, they have to migrate over a long distance, these butterflies, and they orient themselves towards the sun. Extensive studies by Steve Rappert have shown he mounted butterflies on a string so that he could, by video, he could look at their orientation of movements of flight in captivity while exposed to the sun. And he saw that in the northeastern United States, they fly towards their flight movements are oriented towards the southwest. But if you shift their clock by giving them a different light schedule, there is the light advanced by 6 hours, then they also change their direction of movement by about 6 hours, by about 1/4 of the whole scale. And that is proof that the biological clock is being used also in sun compass orientation. That proof has been given for a number of other species, including birds. So the final thing I want to talk about is sleep and wakefulness. Sleep is something which occurs throughout the animal kingdom, and just showing you a few examples. Mice do it, lions do it, geese do it, many different species, and oftentimes you can see from the outside when they do it, when they are active, when they are what is their specialization, whether to be active and wake in the night or in the day. Animals active in the day, so as the European ground squirrel, which you see to the right here, are often camouflaged to protect themselves from visual predators, whereas when they are active only at night, they are oftentimes characteristically colored with black and white colors usually, and they have large ears, large eyes, large whiskers to use other sensory information. And that means that many animals are specialized in living as an active life, either in daytime or at night. And that means that they can over-exercise during that time because there will be another rest time where they are not active, that they can do restorations when they are not active, body restorations, repairs, and in this case in particular on their brain in the inactive time. And that is then either in the night or in the daytime. Now the exact function of sleep is not known, but it's most certainly a restorative function for the brain, perhaps related to changes in the synaptic contacts between neurons. Now, the control of wake and sleep immediately relates to the last part of the talk, the role of circadian pacemakers. The question here is whether the pacemakers generate activity and rest themselves. That has been the common assumption until very recently, that this is the main function of the circadian pacemaker, which is in mammals in the suprachiasmatic nuclei. But there are some arguments against it. And I show a series of these why this is probably not true. And the first point is that when food is periodic or under other conditions, when methamphetamine is given in the drinking water, the SCN is no longer required for an activity arrest rhythm, that even animals without an SCN show this. And also in studies of mutations, that the expression of clock genes in the SCN is not necessary. So that is the first set of arguments, and the other is that the activity arrest rhythm can desynchronize from the SCN. Let's first look at the so-called food entrainment situation. If animals, when the pacemaker is destroyed in the SCN, you can still entrain activity and rest in rats by giving periodic food. Just a limited duration food is being given, and the animals becomes very active preceding the time of when the meal is given. And under some conditions, you can even bring them to free run with this so-called anticipatory activity. So this does not require the pacemaker. Another thing which does not require the pacemaker is the rhythm induced by methamphetamine. This is a rat with where the SCN has been destroyed. It is no longer rhythmic, but as soon as you put a little bit of methamphetamine in the drinking water, the rhythmicity does return and is perfectly running without a pacemaker. Now then, the dissociation between sleep and wakefulness and the pacemaker, that has been found early on already in the early days of Aschoff's experiments. I've shown you this before, the free running situation, but this is what is called, given the name internal desynchronization, is also frequently occurring. And that is a situation where the body temperature rhythm, indicated by these red dots, which are showing the maxima of body temperature, and this is the sleep-wake rhythm, which dissociates from the body temperature rhythm, and forms a peculiar set of movements, oscillates around that pacemaker, but is no longer synchronized by it. When that was known and discussed in the 1980s, my colleague Alex Borbet in Switzerland and I, and Domien Biersma also in Groningen, developed a model to understand this dissociation. And we proposed that sleep and sleepiness have two determinants. First, a homeostatic need, that is the need for sleep, which is based primarily on how long one has slept before and how long one has been awake before. And then in the second place, the phase of the circadian cycle. And the sleep-wake rhythm is simply a build-up of sleepiness in the course of tiredness or of need for sleep in the course of wakefulness, which eventually reaches a threshold and then is followed by a decrease during sleep of this need for further sleep until spontaneous awakening occurs. Now, if this rhythm, this homeostatic rhythm, is operating within an oscillatory bandwidth, then the two get synchronized with each other. But the homeostatic static process will be synchronized, yes, by the circadian system. This will also lead to a phenomenon which is quite interesting, that if people stay awake for 3 days continuously, they will show an oscillation in fatigue, in tiredness. At some points this fatigue will be very high, then it drops again. It is as though they have lost the fatigue, and this is a circadian oscillation superimposed on a gradual increase of fatigue over the 3 days. This is exactly what that model demonstrates, and the model has generally been widely accepted and is widely being used by many researchers to understand sleep and wakefulness in humans. Now, finally, also in small mammals, we have indications that sleep and wakefulness are not strictly controlled by the pacemaker. And in the first place, in recent years, there are data becoming available on what animals do in the field rather than in the lab under controlled conditions. And the first person who finally, after all these years, was able to study Syrian hamsters out in the field observed that in the field, although they are completely night active in the lab, with these very sharp onsets of activity out in the field, they are typically active in the late hours of the afternoon and the early hours of the day, but not in the middle of the night. So the situation in the field is different from the lab. We ourselves have done work on lab mice, which we put out in the field, and we had to go to a facility in Russia, in west of Moscow. There's a facility built by a Swiss researcher, Hans-Peter Lieb, and he allowed us to use that together with him for studies on mice out in the field. And we had mice tagged with transponders there for 2 years in that facility, where they could breed and walk around and visit food feeders. And every time they visited a feeder, they would be recorded. And some mice yielded a complete record over a whole year outside in this facility. And what is interesting is that quite often they showed absolute diurnal activity in the middle of the day rather than in the night only, as we know from lab studies. And this holds also for the population as a whole. There were in total of these 2 years, 163 lab mice have been recorded, and here's a summary of their joint activity, and that is quite frequently the activity is in the middle of the day rather than at night. So as though we are being put on the wrong foot by looking at lab studies alone. The lab is an unusual situation. For the field is totally different, and the main difference between them is that in the field, animals have to work for their food, to obtain food, and to survive. And in the lab, the food is always continuously present and close by and easy to get, and they don't have to work for it. So we thought after these experiences and the difference in the activity patterns that we might better look in the lab at a situation where mice have to work for their food. That's easily done because you can reward them for every couple of rotations of their wheel. You can give them a food pellet, and then you can increase the decrease the reward rate and thereby increase the workload. And that is exactly what my then student and now colleague Roelof Hut has done, letting mice work for their food. And it's very clear that if you do that, that they shift their activity over from the night into the day. And if you then release them in continuous darkness, it turns out that their activity starts from where the night began before. So the pacemaker, which takes over control again, is shown not to be moved at all. So the activity is moved relative to the pacemaker. And also, if you give at the same time a temperature recorder in these, put a temperature recording sensor in these mice, then you see that they let drop their body temperature during these in this situation to very low values, around 21 degrees, and they can easily sustain that. Humans couldn't do that, but animals can do that. So in this way, when food is scarce, mice can save a great deal of energy by moving their activity to the warmest part of the cycle and cool down during the night. And that is a thing which is highly adaptive, this flexibility of sleep and wakefulness relative to the external cycle, and it is not dictated continuously by the pacemaker. Pacemaker exerts loose control over wake and sleep. And that is summarized here in this slide. We have light and darkness which enter the eyes, which then that information is transferred to the pacemaker in the suprachiasmatic nuclei, the SCN, and the SCN acts as a synchronizer. It gives an internal representation of external time to which different oscillators in the body temperature, activity, rest, hormones, the nervous system can listen to adjust to their optimal time. That control is flexible. So in summary then of what I have told you is that circadian rhythms are a property of life on Earth which is nearly as ubiquitous and as fundamental as DNA itself is. It is everywhere. And circadian pacemakers in the central nervous system provide animals with a cycle of internal time synchronized with the Earth's external time through light, following very basic functional rules, the rules summarized in the phase response curve. And they usually synchronize physiological periodicities throughout the body, which subserve a great variety of functions. And with that, I think I end my talk. It has been long enough, and I hope you have enjoyed this. And with that, I hand over to Matthew Cobham. Thanks very much.
So thank you very much indeed, Professor Daan, for that fascinating talk. Here we have actually the email address where you can send your questions in. And I would also like to highlight some things to you before we finish today's webinar. The first is to mention about up-and-coming webinars, which are now in planning for the autumn, September, October, November, and December. And you will all be sent information on that in the coming weeks as it becomes available. The other thing to mention is an upcoming event which is being planned for early November. It was also mentioned in the last webinar, Luminous Talks again in collaboration with the Parsons New School for Design. And again, we will inform you about this, but also if you would already like to express an interest in being involved in that, please send an email to the same email address as where you can pose questions to Serge Daan. The event will be held at Parsons, the New School for Design in New York, and yeah, just to mention again, at the beginning of November. So thank you very much indeed to everybody, and yeah, on behalf of Philips and the Philips Lighting University, I'd like to thank you for joining, and please forward any questions you might have.